well. So today is, what is today? Friday, April 24th, and that is way too small. This is going to be our histology review. I am glad some of you were able to make it. I really liked the idea of doing this. So after I spoke with you guys on Wednesday, I spoke with my 4:30 class on Thursday and decided to do something similar with them. And as I spoke with you guys and we were talking about, one of the things that I wanted to do was be able to allow this for extra credit. But I appreciate that this is outside of class time, so that was a bit of a conundrum. So in discussing this with them, what I realized, which was a great win-win for everybody, is that this being here for the review is not extra credit, although I strongly encourage you to be here so that you can ask the questions and make sure that you are getting out of this what you need. But for extra credit, what you are going to do is actually complete the histology images. So there's the histology portion of the handout from your study guide. If you remember way back to 4:30, on the very first one where we did the epithelial tissues and connective tissues, you had to draw the drawings, label the drawings. Color would be nice, not required. But if you do all of that and complete it and turn it in, I've already set up an assignment on Canvas for us. If you do that, it is going to be worth 10 points of extra credit. So again, this is not a required activity. You're not required to be here, but the histology is important, especially with the digestive system. There is a fair amount of it and it's fairly diverse. So I think that this is worthwhile. And so that'll be due. And because we didn't get a chance to talk about it in class, I have decided that it will be due by the beginning of the exam. So this way, those people who weren't here or don't get the opportunity to look at this don't lose out on that opportunity. So I'll let them know on Monday. So it's going to be due Wednesday. Oops, what just happened? This is going to be due Wednesday, and that will be April, what is that, the 29th? By 7 a.m., because that's, I believe, when our exam is. Is that correct?
Yeah, I believe so.
Okay, excellent, excellent. Yes, I am recording this. I will still be posting this on YouTube. So like I said, I guess if you truly need to go back to sleep, you are welcome to do that. But as you always know, coming to class, being there, being able to ask the questions, make sure this is clear for you is better than coming back and reviewing it later. But yes, for those that missed out, for those, and again, because this is outside, that was my real conundrum with this, is because this is outside the normal class time, it's hard to make it a required activity. So I didn't want to make it a required activity, so I will record it to give people the opportunity and the opportunity to do this. And again, the whole point of this is a little carrot to give out to you, but really it's to help you with this material. All right, so that's going to be the game plan. Questions on any of that? All righty, excellent. So let's go ahead and switch. Stop sharing this now that we know that. And then I can. There you go. We're going to do this in a very similar fashion to what we would do in the classroom. And that is I show the slide on the screen and we talk about it. Again, these are unfortunately static images because I do not have my own microscope at home. And so I did just the same thing you should be doing as you're studying this thing as well. I did a Google search to try to be able to find the things that I was looking for. Again, you have the advantage also of using your other materials that you are responsible, what you have, your a professional anatomy lab, you have your textbook, your lab manual, hopefully you have a histology atlas. All of those are things that you are responsible for and have and should be helping you on this. Now, what we have here is the first item on the list, and we're going to go down it through as the way we always do. This is indeed a tongue. We can see it is a relatively low magnification view of the tongue, where we can see up here the some of the irregularities and papillae of the tongue that we see. If we were to look closely, and we'll do that in a moment, we could see that this, of course, is that non-keratinized stratified squamous epithelial tissue. Being in your oral cavity, do you use these? So yeah, possibly. Often when making the lab exams, I use images from your textbook, images from your atlases, images from your other resources, because we do get a database of that. But often if I'm trying to do something, I will go to the almighty Google. As I've said many times, my goal for you is to learn this material, not to memorize the pictures in the textbook. So while I think pictures in your textbook and pictures in your histology atlas are a great starting point for these things, it is definitely important to look at these things and be able to recognize and identify those things. And the nice things about your histology atlas in your textbook is they should be nice, good, obvious examples of them. But it looks a different way, different stains, different magnifications, different planes of sections. All of this stuff looks different. So as I've said many times, look at the histology book first, look at your PAL CD first, do all of those things first. And then as you start getting comfortable, go to the almighty Google so that you can see all the variety of it as well. Right? It's hard to start with that Google because if you don't know what you're looking at, it's hard to tell what's correct and what isn't correct because things are often mislabeled or often missearched when you go through them. But as you get more comfortable with them, then it can be. So yeah, I have no problem using a novel image. I will say that none of the images that I've got here today are images that I have pulled from our exam. Again, these are things that I specifically got novel just by going on to Google, but could they end up on the exam? Sure, or I'll probably pick something else as well. Again, I'm not going to limit myself that way. As long as it's a good example. All right, so here we have our tongue. Our tongue, of course, is in the oral cavity, open to the outside world. So, of course, this is lined with a mucous membrane. So, of course, this is our epithelial tissue. And as we mentioned with the tongue, it is a non-keratinized stratified squamous epithelial tissue. And it sits on top of an areolar connective tissue. And of course, because this is a mucous membrane, this areolar connective tissue is a lamina propria based on its location. Notice a couple things at this low magnification view we can see. As we know, the tongue is a muscular organ. And one of the unique characteristics about the tongue is the tremendous disorganization to the fascicles. We see lots of bundles of muscle cells at all sorts of different orientations, some more longitudinal sections, some in a complete cross section, some in more an oblique section. So we have a bunch of that. The other thing we see, and notice we see it up here in the lamina propria, but we also see it deep in the tongue as well, are these mucus producing glands. So there's all these mucus producing glands located within this space as well. The other thing I liked about this is because what we can start to see, if you remember we were talking about, are some of the papillae that you are going to be responsible for. We did mention how the surface of the tongue is lined with 4 papillae. The lateral ones, the foliate papillae, are not something we can really see on a microscopic view. I mean, I guess maybe technically if you took I guess that would be I would say sagittal would probably be in a more appropriate term, section through, no, it wouldn't be sagittal, maybe a cross section through the tongue or something like that, where you were able to go from lateral edge to lateral edge. You may be able to see it, but that's typically not how you see them. But what we do see on this is 2 of the types of papillae. The first are our filiform papillae. Remember, the filiform papillae are going to be more narrow, They're more sharp looking at the edge of them. These are the ones that, remember, provide the tactile sensation, the temperature sensation, things along those lines. But then here, what we see is this massive papillae kind of embedded within the surface of the tongue. And this is that circumvallate papillae. Notice this circumvallate papillae also, and again, we can't increase the magnification of this, but I do have another view that will show something similar. Notice there are these light spots along the lateral aspect of the circumvallate papillae. We see them here and we see them there. Those structures are actually the taste buds. So this tiny little structure that we see, and again, I will show you a bit of example, but all of these things right in here, these are the taste buds that are on the lateral edge of our circumvallate papillae. And one of the big differences between the circumvallate papillae and a fungiform papillae, A, is the size, but B, also the circumvallate will sometimes have the taste buds on the apical surface of this as well. And that is, this particular one doesn't show that. Again, maybe a different section of it would, but we clearly see those taste buds along the edges of it here and along the edges of it here. All right.
Quick question.
Yes, go ahead.
Are the fungiform and circumvallate, are those interchangeable terms, or?
No, they're two completely different types of papillae. Remember, if we think of the tongue, and I'll just draw a real simple one there. Remember, there was that line of prominent papillae that kind of helped to form the boundary between the anterior 2/3 and the posterior third of the tongue, right? There were a bunch of little circle-like structures that we saw all kind of in a row like this. These are the circumvallate. That's what we're seeing here, this big, huge, massive. The fungiform are these small little mushroom cap ones that are found throughout the surface of the tongue.
Can the circumvallate, can the circumvallate be shortened to just vallate?
Yes, circumvallate and vallate are interchangeable.
Okay,
circumvallate and fungiform are not. Yeah. Yeah, like I said, circumvallate is the appropriate term, but I've seen more and more in the literature that they're going more and more into just calling it vallate papillae. I don't know why, but it's being done. And so, yeah, I'm fine with vallate as well as circumvallate. Both are acceptable terms. All right. Let's take a closer look. This one, on the other hand, is well, here, again, another circumvallate papillae. Again, we can see it has got these deep invaginations, deep below the level, big, huge. And again, here we more clearly see those taste buds on the lateral aspect of it. There are none on the top there, but we see those lateral aspect. These light colored structures are the taste buds. Taste buds are actually kind of a bulb-like structure. It's kind of like a cup-like structure that is opening, and it has an opening at the top where the chemical can come in. And then inside of it, it has the cells that are capable of perceiving that taste sensation and carrying that taste sensation to our brain. One of the interesting things about this is the taste buds themselves only last for 7 to 10 days. Which isn't surprising if you think of all the toxic things you put in your mouth, that hot coffee, that, spicy tortilla chip, whatever it is that you're putting in there, these get damaged and they get replaced frequently. If you're a chronic smoker, these things get damaged. But if you stop smoking after a couple weeks or even a month, depending on how long, your taste sensation comes back better as a result of that. So we see those nice and prominently along the lateral aspect of, again, this big, huge circumvallate papillae that is kind of anchored deep into the tissue. Notice also here, we can again very clearly see that non-keratinized stratified squamous epithelial tissue, both on the surface of the tongue and on the surface of the circumvallate papillae. And then again, here we have the areolar connective tissue. And then deeper down in here, we have, like I said, we can see some glands that are labeled here as well. Yes, you are responsible for them, even if they're not on the histology list. They may not be on your histology list, but they are on your anatomy list. So if you go up to your anatomy list, I would be shocked if they're not up there. Yeah, they are on your tongue list for your gross anatomy, and they are something that can be seen with the naked eye. So that's why they aren't necessarily down here on the histology list, because they're something that can be seen as the naked eye, but you will also be responsible for identifying them histologically as well. So thank you for asking that. So it is technically on your list, but you're right, it isn't listed in the histology just because it is actually something I can see with the naked eye as well. Now, here, on the other hand, are some examples of the fungiform papillae. Notice the fungiform papillae, as their name indicates, kind of fungiform, fungi, mushroom cap type space. They stick out. Rather than being embedded within the surface of the tongue, these stick out. And again, notice we very clearly see these light colored circles on the sides. These are the taste buds. And in fact, if you look closely right here, you can actually see that pore. That is the pore where the chemical dissolved in water, dissolved in your saliva, comes in and then gets perceived by those taste sensors. So you can see a clear pore there, and you can also see the opening of the pore on that one as well. So we've got a really nice section of this one where you can very clearly see that pore into that taste bud. Whoops. All right. Much smaller in size. If you think of the relationship of the size of the taste buds to the papillae on this one, right? Even if we wanted to, we probably couldn't get 6 taste buds here along the size of our fungiform papillae, whereas clearly we have more than a dozen here along the side of the circumvallate. Again, the circumvallate is so, really, the fungiform can be seen with the naked eye as well, but these are a much larger, much more prominent structure. And the relationship of the size of the taste buds to the size of the papillae should be one of the easiest ways to tell the difference between the circumvallate and the fungiform papillae. Lastly, here we see a really nice classic example of the filiform papillae. My guess, based on the look of this is probably actually a cat tongue. Remember, we talked about how that cats have a much more prominent filiform papillae, given their tongues that very rough texture to them. Sometimes they can be more subtle, although this is really more of a plane of section probably type of thing. But notice the other key is nowhere along the surface of this anywhere do you see any of those taste buds. Because remember, the filiform papillae give us things like tactile sensation, give us things like temperature, but they do not give us taste. And again, notice here we see the beautifulness of the different variations in the orientations of the fascicles. And again, notice as we look at one of these muscles, there's no way for us to tell whether this one is extrinsic and this one is intrinsic, right? So I'm not going to point at one of these and ask you, is this an intrinsic or extrinsic muscle? But remember, you are responsible for being able to know the functions. You know that the intrinsic muscles of the tongue change the shape of the tongue, and the extrinsic muscles of the tongue are what move it through space. All right, as I look at my list, I think that is it for the tongue. So any questions on the tongue? All right, silence always golden. We did this one in lab, but again, there are still some things we wanted to look at this again. So notice here, we see our esophagus. The plane of section of this esophagus is what? What would the plane of section of this one be?
Transverse.
Right, transverse or cross section, absolutely. Notice again, it has a similar anatomy to our basic digestive system anatomy. We have a mucosa that lines the surface of it, and that mucosa has an epithelial tissue, a connective tissue. And notice here we actually do have a minor muscularis mucosa. So we actually have a little bit of a muscularis mucosa on this one here as well. That's nice because it helps to distinguish it from the submucosa. Here's our two layers of muscle. And again, the one closest to the lumen is going to be circular. The one furthest from the lumen is going to be the longitudinal, right? So all of that, this we know is a retroperitoneal organ. So we would just have an adventitia on the outer surface here. This one doesn't show it very well, but it would be out here on the outer surface. And the big difference, as we talked about, is that this, instead of having a simple columnar epithelial tissue, has a non-keratinized stratified squamous epithelial tissue. Notice also this is basically a collapsed muscular tube, so there are folds in it. I will say this fold is similar to what we would think a rugae would be in the stomach. Notice that both the mucosa and the submucosa follow and form that, but really, for whatever reason, I don't have a good reason as to why that is, they're not referred to as rugae, but essentially it's the exact same type of thing. It is a fold in the mucosa and submucosa that is going to allow this to expand to accommodate your bolus of food as you swallow it down. Right? Unlike the trachea, which we want to keep open all the time, the esophagus is only open when food is passing through it. So it's this muscular tube that collapses, and when it collapses down, you get these folds, these invaginations in the mucosa and submucosa, right? As I said, this is very clearly a cross section, and equally such, this is very clearly a longitudinal section. Notice we can still see the same basic anatomy in this picture. Again, I was lazy with some of these things. As you can see, I stole stock photos from things, so you see the watermark on them. This one happened to be labeled, but it serves our purpose, so it was fine that way. Notice again, we can clearly see our mucous membrane, our mucosa, made up of our non-keratinized stratified squamous epithelial tissue. Our lamina propria, which is the real connective tissue, our muscularis mucosa, our submucosa, where we find the large blood vessels, larger glands would be found in this area as well, although I don't see any in this picture, a little bit adipose. Our circular muscle layer, and our longitudinal. And again, notice clearly these are perpendicular. We can see this is a cross section through the fascicles here, whereas this is more of a longitudinal section. And again, we see that collapsed muscular tube. So it should be pretty easy to distinguish a longitudinal section from a cross section, and that is one of the things you are responsible for doing. However, the other thing that I wanted to show you on this, and we looked at the one picture, but I got a couple more of it as well, is notice here we see that great transition region. Remember, as we talked about 2/3 of the way down the human esophagus, it transitions abruptly from a non-keratinized stratified squamous epithelial tissue to a simple columnar. And again, that transition is incredibly abrupt. Here we see a great example of it. Here we see another great example. Now this one happens to be in a rat. One of the interesting things about the rats is that transition actually occurs at the opening of the stomach, where the esophagus feeds into the stomach, and where our cardioesophageal sphincter or lower esophageal sphincter or gastroesophageal sphincter is located. But the point is, again, I wanted to show you that that transition of epithelial tissues is abrupt. So it's kind of cool that way. The other thing that I told you about, is we've been talking about it lots of times, is these sphincters, how they are enlargements of the circular layer of the muscularis externa. And I found this picture here, which does a great job of showing this. Notice this one does appear to be human. We can tell that because we can see up here in the esophagus, it is already a simple columnar epithelial tissue that is lining its surface here. And here is the beginning of our stomach. And notice here we can see our longitudinal layer of the muscle, which is this small thin layer right here. And here's our circular muscle that's coming in right here. And same thing here. Here is our circular muscle and our longitudinal muscle. And you can see this massive enlargement of the circular muscle, which is forming that sphincter. In this case, this would be that lower esophageal, or like I said, cardioesophageal sphincter, basically bounding the opening of the stomach and separating it from the esophagus. So here we get a really great view of how big and large that a big circular layer can become to become that big, huge, large valve that is going to regulate the flow out of that conduit that is the esophagus. So again, it's the lower esophageal, cardioesophageal, gastroesophageal. All of those are acceptable terms for that. And that's kind of it for that. Again, not a whole lot with the esophagus. Questions on that? Alrighty then. Oops. Oops, going the wrong direction. There we go. Excellent. From this then, the next thing on our list is something that we already looked at, but I will look at it again. And these are our salivary glands. We have 3 pairs of salivary glands. Each of them are different by the composition of their glandular structures, and therefore we can distinguish what they make and which ones they are. Which salivary gland is this one? Which of our 3 salivary glands are we looking at here? You guys all asleep already?
No, just done.
Well, you should have your histology list in front of you, and seeing as we've been going in order, what would your best guess be?
Parotid.
There you go, excellent, right? And you could have either figured that out because it is the first one on the list, or the key to identifying your salivary glands is to pay attention to the secretory structures. Remember, all glands, and this is important to remember, all exocrine glands have 2 parts. That's way too big. Right? All exocrine glands have 2 main components. All right? The first are the ducts. Those are typically made up of simple cuboidal or columnar, although they can be bigger than that. But those are typically what they are, and they're pretty easy to tell. A duct is a duct is a duct, and they're pretty much the same in every gland you're ever going to see. However, the other component of an exocrine gland are the secretory structures. And one of the important things to remember, as I'm sure you recall from 430, and if not, when you go back and look at epithelial tissues in 430, there are basically 2 main shapes to our secretory structures. Those are the acini, which are ball-shaped, and there are the tubules. Oops, so I spell that right. That are tube-shaped. And again, that is true for all exocrine glands. They're going to be made up of some type of duct, 1 or more ducts, and some particular shape of secretory structures. All right. Now, when it comes to our salivary glands, saliva has 2 main components to it. It has a serous fluid. This is the watery part of it. This is the part that contains the antibodies, it contains the enzymes, and contains the defensins and all the other fun stuff in there. But it also has a mucus component, right? Or really something that produces mucin. So if we're going to make saliva, we need something to make serous fluid, and we need something to make mucus, something to make mucin. Conveniently enough, we have 2 different shapes to our secretory structures. So different secretory structures can make different stuff. And in the case of the salivary gland, it is the acini that produce the serous fluid, and it is the tubules that produce the mucin. All right. Now, if you remember when we've seen goblet cells, when we've seen other things that way, we know that mucin, most stain doesn't bind to it. So when we looked at the secretory structures, they tend to be fairly light in color or clear in color, kind of look big and pillowy, similar to adipose, but not exactly like adipose, because remember, adipose shoves the nucleus way to the side. The mucin doesn't do that the same way, but it does have that same kind of clear appearance to it that adipose does, and like a goblet cell. As you look at these secretory structures, do they look light and clear and pillowy?
No.
No. They also are all fairly similar in shape. They're small, tight, ball-shaped structures. There's not a lot of variation in the shape of these secretory structures. Because again, when you have a ball, no matter how you cut the ball, it's still pretty much a round-shaped structure. So the uniformity of the secretory structures screams acini. The fact that they're dark in color screams not mucus, therefore serous. So these are serous producing acini. These are ball-shaped secretory structures that produce serous fluid. All right? And they're as far as the eye can see. So all we have are serous producing acini and some ducts. So based on that, which of our 3 salivary glands is this one likely to be?
Parotid.
Yeah, it's still the parotid. We haven't changed the slide. Absolutely. Is our parotid gland. Remember, a parotid gland, that big, huge one up on your cheekbone. And again, not only, think of it this way also, parotid gland produces the serous fluid, which has, like we said, it has the watery components, it has the enzymes, it has the antibodies, it has the defensins and things along those lines. The other thing it has in it is some of the buffers. Right? If you think about it, when you eat something really, really sour, you sometimes get this kind of sucking sensation in the sides of your cheeks. That is that feel of the saliva being drawn through those parotid ducts into your oral cavity to help to neutralize the acidity of that sour lemon or whatever it is that you're sucking on, right? So you get, sometimes when you eat that sour stuff, you get that sucking sensation in the sides of your cheeks. That's because of the activity of your parotid gland. All right. Now, with all glands, we need to not only just identify this gland, but we need to know its structural classification, its functional classification, and what it produces. Of course, we've already said what it produces, but tell it to me anyway. What is it that it produces?
Serous fluid.
Yeah, serous fluid, the watery component of saliva. Both of those would be perfectly acceptable ways of describing that. What is its functional classification? What is its mode of secretion?
Merocrine.
Merocrine. In fact, every digestive gland uses the merocrine mode of secretion. So that makes that question really easy. You see me write what is the functional classification of this gland on this exam, and you don't really even have to look at the picture, right? Because it's going to use the merocrine mode of secretion, exocytosis. There aren't any that use holocrine in the digestive system. So that makes it super easy, right? So then what that leaves us with is the structural classification, right?
Compound acinar.
Say again?
Compound acinar.
There you go, compound acinar. Remember, compound means 2 or more ducts, 2 or more secretory structures. We clearly see at 3 ducts right here, uno, dos, tres. So really, and then obviously far more than 1 secretory structure. So absolutely, this is a compound acinar. And again, remember, I keep using acinar for consistency, but remember the term of velar is also perfectly acceptable. Both are acceptable terms for this. Excellent. So what's this? Identify this gland.
Sublingual gland,
There you go. Excellent. How did you know?
because it's light colored.
Excellent. And it's also next on the list, which is also convenient. But you're absolutely correct. Notice, once again, ducts are ducks are ducts. Here's a small one. Here's a small one. Here's a bigger one. So we're clearly in a gland. So what we look at next are the secretory structures. And as we look at the secretory structures, there are, again, two characteristics that help us distinguish them. Color is not something we always want to pay attention to, but sometimes it is important. And clearly, these are very light in color. They're very light in color because they primarily contain mucin. And like we said, there are some stains that will bind to mucin, but most stains don't. So typically, goblet cells, mucin-producing cells are very clear in color. Most stains don't bind to it, and so that's what we see here. The other big difference, even if we didn't pay attention to the color, the other big difference that we see here is there's less uniformity in the shape. Again, remember our two things are tube shape and ball shape. No matter how you cut a ball, it looks the same, right? With an apple, if you cut an apple, pretty much most ways you cut an apple, it's going to have pretty much a bit of a round shape to it, and it's not going to look that much different. Is the same thing true with a banana? Can you cut a banana every which way and they all look the same? No, if you cut it in a cross section, it looks completely circular. If you do a sagittal section, it looks like a smiley face. An oblique section, it's going to look like an oval, right? There's all these different ways to cut that banana and it's going to look different. And it's the same thing here. There's all these different ways you can cut a tube. Sure, some of these tubes are cut in complete cross section, and they're going to have a nice circular shape to them. Like this one's almost a perfect circle, and you can actually perfectly see the lumen at the center of the secretory structure. But notice this one's here more cut in a longitudinal section. Again, we can see the lumen at the center of the secretory structure in a longitudinal section. This one's oblique. Here we're cut it on a curve. We got one of the curved parts of it. Here's kind of oblique, more closer to cross section. There's much less uniformity in the shape of these because when you're dealing with tubes, when you cut them randomly through them, you're going to cut them at different shapes. So we have that consistency of color, and it's a light color that tends to tell us mucus. The fact that we have more irregularity in the shape of the secretory structures tell us tubules. So clearly this is a gland that is comprised almost exclusively of mucus-producing tubules, producing a tremendous amount of mucin to give the mucus quality to our saliva. And of course, which gland is that? I'm sorry, which one?
Compound tubular.
True, absolutely. That is its structural classification. Absolutely. Structural classification is a compound tubular gland. And like I said, that one that is the compound tubular gland is the sublingual gland. And if you think of where that's located, that's the one that has the multiple ducts that feed out of it. Really short ducts. It's right under the tongue. So if we're producing this thick, viscous mucus, it doesn't have to travel as far, and there are lots of ducts to get it where we need it to go. All right. And that brings us here. Once again, we see ducts, lots of ducts in this case, and we see secretory structures. But notice, unlike the previous two slides, our secretory structures are not uniform. Some of them are dark, compact, circular structures, and some of them are these weird-shaped pillowy structures. That clearly means we have a mix of both serous acini and mucus tubules. So this gland is producing a mixed saliva of both serous fluid and mucin. And which one would this be?
Submandibular.
Submandibular.
Submandibular. Submandibular, excellent. And then the only tricky part on this one, again, remember they all use the American mode of secretion, but what is the structural classification of this one?
Compound tubular cinar.
There you go. It's got the mouthful word. Compound tubular acinar, because it has both tubes and acini. And there you go. So you notice if you pay attention to the acini, it's pretty easy to tell these apart histologically. It shouldn't be that challenging. It should be pretty simple and straightforward. All right. Questions on that? All righty. With that, then we are done with the salivary glands. Let's move on to the stomach. Now, here we have a nice low magnification view of the stomach. And here, notice this damn near looks exactly the same as what we saw in our esophagus, where we have our mucous membrane, our mucosa, we have our submucosa forming this big fold that will allow it to expand. However, because we're in the stomach, what do we call this structure here?
Rugae?
Yeah, this is the rugae. Absolutely. Now, there are still some ways that we can tell that this is the stomach and not the esophagus. One of those is down here. Notice as we look down here, we see 3 layers to our muscularis. We see our longitudinal, we see our circular, and then we see portions of our oblique layer up here as well. The other way we can tell this is the stomach is by looking at the mucosa. Notice the mucosa is very, very thick, much more extensive. And even at this low magnification view, notice we can see those gastric pits. Remember, the gastric pits are the invaginations of just the mucosa. Notice we can clearly see the muscularis mucosa. So from here up, all of this is the mucosa. So here we have these invaginations of the mucosa, and the dark distinct top parts of them are those gastric pits. They are going to feed into the gastric glands, and that's what we're seeing here. We're not really seeing it at a cellular level, but we can clearly see that this is going to be those gastric pits then in here, and we'll talk about the significance of that in just a second. So we have that. We have a little bit of mucus even on the outside there, lining the surface of that there. And like I said, the muscularis. And then here we have the teeniest bit of that serosa that would be on the outer surface. Oops, wrong, come back. All right. Now, one of your jobs, and I'll be honest, I had to throw this together relatively quickly, and I did not find a slide that I loved. Not only do you need to distinguish the stomach from the other organs, but as we talked about, there are some distinct differences between the regions of the stomach. I am not going to hold you responsible for distinguishing all the regions of the stomach, but the one region that is most specialized is the pyloric region. So what you will be responsible for is distinguishing the pyloric region from the rest of the stomach. In fact, if you look at the histology list, you see it written that way. I have stomach, and then the next slide is pyloric stomach. So what we are looking at here is likely part of the fundus. It might be the body, but it's hard to tell from this view. But here are the 2 keys that you're going to be looking for to be able to tell them apart. The first is the size of the muscularis. Remember, one of the things that I told you about the pyloric region is it is much more muscular. All right, so this here, we see the entire muscularis in this. And again, yeah, I don't have anything to compare it to, but I will tell you this is relatively small-ish, although it can be much, much thinner, especially up in the cardial region. But the other way it's going to be different is in the composition of the gastric glands. The gastric glands in the fundus, in the body, have more of the parietal cells, have more of the chief cells. So they have a darker coloration to it. Even though we can't see the cells here, the fact that the pyloric region has a larger muscular area and has more mucus-producing glands. And mucus-producing glands, of course, have mucin, and so they're much more clear in color. So the fact that this is dark in color tells us that this is not the pylorus. All right? Here we see a thicker muscularis. This is actually the body when we're close to the fundus. Pardon me, not close to fundus, close to the pylorus, but we're not quite to the pylorus yet. Notice the muscularis is getting larger. We can clearly see the 3 layers to it by the difference in the coloration, the difference in the orientation of the muscle layers. And again, notice we can see the gastric pits up here at the top, fairly uniform, and then here, the different colors of the cells in the gastric glands. And again, these gastric glands are just a component of the mucosa. So again, we have that muscularis mucosa, and then our submucosa, and our muscularis and serosa. Here, we get a little bit of a closer look, and again, this is the fundal region. Again, we can see those simple columnar cells very clear in them. We can see some of the mucin now that is in them. These are those goblet cells. These are these mucus-producing cells that line the surface of the stomach and line the gastric pits. But notice we don't see that same type of goblet cell-looking appearance to the cells down here. Now, as I mentioned, is it possible to look at one of these particular cells and be able to tell what it is? Yes. Do I expect you to be able to do it? No. So just know that these are the gastric glands. Know what the 4 gastric gland cells are. Know what the 4 gastric gland cells do. But I'm not going to make you recognize them histologically. This is what the mucosa looks like in the stomach of our fundus. This is what the mucosa looks like in the pyloric region. Notice we still have those gastric pits that we can see on the top, but notice now when we go deep into the gastric glands, as we go deep into the gastric glands, we see almost exclusively light-colored clear cells, and these are those mucus-producing cells that are here in the pyloric region. Whoops. I couldn't find a view that did a nice job of showing that huge, massive thickness to the muscularis, but your textbook might have it. There might be other resources. I purposely, when putting this list together, went to Google to get them because I didn't want to show you the same pictures that were on your histology atlas or in your textbook or in your lab manual because I figured you already had those to look at. What I will tell you is while putting together both your list and the list for, because I'm doing something similar for the 430 class, what I did find was, I think practically a third of the pictures, if not more, the pictures I found were from this amazing site from Yale University. So if you've looked under the study tools, under the modules on our Canvas site, you will see that I've added that Yale Histology website, which is really amazing. It shows really great images. You can look at it with and without labels. It is an extensive list. I looked at it a little bit because, like I said, I was short on time when putting this together, but it looks amazing. So I would encourage you to spend some time on that side as well because it is really, really beautiful. And many of the pictures I have came from it. But that's really the one of the big differences, right? You can clearly see this and that are very, very different from each other. So even if you can't see the muscularis, the composition of the gastric glands should be sufficient to help you to be able to distinguish the 2 portions of the stomach you're responsible for, the pyloric region and the non-pyloric region. All righty. Questions on that? Excellent. All right, so on to the small intestine. We haven't talked about this, so we're getting a little ahead of ourselves. Well, actually, we did talk about a little bit of this, but we are still starting to get a little ahead of ourselves as we move forward from this. But I still think it's worthwhile to take the time to go over these things. One of the things that we already discussed at the end of the last class is the anatomy of the small intestine, and how that anatomy of the small intestine has these circular folds inside of it, which are essentially like the rugae, made up of mucosa and submucosa. But along the surfaces of those rugae, we then have the villi that stick out. And on those villi, we then have the absorptive cells that have their microvilli on the surface of those. So layers upon layers upon layers of specializations to increase the surface area, like I said, close to 600 times. What we're looking at here are those villi. And again, these are characteristics that all portions of our small intestine have. They all have villi. They all have a submucosa and a mucosa and a muscularis and all of those things. And so it can be challenging to tell them apart. The size of the villi will vary. Right? Remember, we said the majority of the absorption is occurring in the proximal portion. And the duodenum is very, very short. So the ileum tends to have the longest villi, followed then by the duodenum, followed then by the jejunum. But especially if you're just looking at one picture by itself, these look pretty long to me, so I don't know necessarily which one it is. As we've talked about many times, the key to quick and easy success in identifying things histologically is knowing the key. Usually there's those dead giveaways. And when we're dealing with the small intestine, the dead giveaway is going to be the submucosa. As soon as you see villi like these, then the first thing you should do is look in the submucosa. Notice here's the villi. Notice we see some goblet cells on the surface. There's clear light, nice and obvious and distinct. Here's our muscularis mucosa, and boom. Here's what we're looking at underneath. Here's our submucosa. And as you can see here, and this is the way you're going to be able to identify it, are some glands. Massive, massive mucus-producing, bicarbonate-producing glands here in our submucosa. These glands are sometimes referred to as Brunner's glands, because good old Bob Brunner was the first person who described it and stamped his flag on it. But the other thing these are known as are duodenal glands. And guess what specific region of the small intestine you find duodenal glands in?
Duodenum.
Yeah, in the duodenum, absolutely. So if you look in the submucosa and see duodenal glands or Brunner's glands, you know you're in the duodenum or duodenum. Notice here, this one's a nice cross-section view. We again have extensive villi. Those villi happen to have goblet cells on them. All right. We have our two layers of our muscularis. Right, a little bit of serosa on the outer surface. But notice here, here's a tiny submucosa. It looks like we've got a little bit of a blood vessel in here. That looks like actually it might be a nerve, right? But there's not a whole lot going on in the submucosa here. Extensive villi, now by comparison, we can see these are longer than the ones before. But notice here, again, our goal is look in the submucosa. You don't see duodenal glands, right? You really don't see anything. Let's look at this one by comparison. Notice here, our villi are still pretty extensive. They may be a little smaller, but again, it's subjective. It's hard to say. Here's our muscularis. Here's our submucosa. And notice here in our submucosa, we have these big, huge, round, dark stained things. Any idea what these big, huge, round, dark stained things might be?
The
Pyrus
prostate-
patch.
Yeah, how about Pyrus Patches? It says it right here, absolutely, right? Those lymphatic follicles that we talked about. And what region of the small intestine did we say we would find the Pyrus Patches in?
Ileum.
In the ileum, right? It's the one closest to the large intestine. And the large intestine is where that huge bacterial flora and fauna is. So we need these bodyguards to protect us here in our ileum from any of those bacteria that might go wandering. So notice the submucosa of our ileum is going to have Pyrus patches. The submucosa of our duodenum is going to have duodenal glands. And the submucosa of our ileum has neither. So notice the way we're going to tell the three. So notice again, we just look up here apically. I see villi, I see villi, I see villi. Maybe this is the longest, maybe that's second. Again, the magnification's slightly off, so it's hard to tell, all right? So villi can be a way to distinguish them, but it's more subjective. What's not subjective is what's going on in the submucosa. You either have Pyrus patches, you have duodenal glands, or you have nothing. It is a clear, obvious way to tell the three regions of the small intestine apart histologically. All righty. Questions on that? All right. From here, then, we are truly into the unknown, things that we have not had a chance to talk about yet, but I still think it's worthwhile to at least look at so that it can help us move through it more quickly as we move on. This is our colon, our large intestine. Notice, doesn't have villi. It does have big intestinal crypts. And notice these intestinal crypts along the entire surface are filled with goblet cells. Pretty much the main secretion that is produced by our large intestine is mucus to help grease the wheels to move that what's going to eventually be feces out of our body. Right? By the time our food has reached the large intestine, most of the processing has been done. So really, it's about extracting the last little bit of nutrients, letting the bacteria do that work so we can get a little bit of nutrients out of it, withdrawing excess water, right, additional water. Remember, most of the water is pulled out in the small intestine, and packing it and greasing it so that it's easier for expulsion. This can look a little bit like the pyloric region of the stomach, but what I would say is that there's two ways to really tell them. The first is that the mucosa of the pyloric region is much more massive, huge thickness to it. These intestinal crypts are not nearly as elaborate as those gastric glands are. But again, we always want obvious things, and the obvious thing is this right here. How many muscle layers do you clearly see here?
2.
2. And if you see 2, are you in the stomach?
No.
No, because remember the stomach has 2 muscle layers. Pardon me, 3 muscle layers. So the fact that we see 2 here tells us we're clearly not in the stomach. So all the mucus glands may make us think pyloric region. The 2 muscle layers clearly tell us, boom, this is the large intestine. This is the colon. All right. I think that's it for that. I don't think there's a ton we need to know on the colon. Obviously,
What are those circular things in between the top and the bottom?
this?
Yeah.
I don't know. I've never seen that before in my life. No, I'm just kidding. Anybody want to hazard a guess? What does it look like? What does it look like they're filled with?
Mucus?
No, not a bad guess. That is an excellent guess. Those are actually red blood cells. This right here, if you look at the size of the muscularis, is a vein, and this is an artery. Notice it has a thicker muscular wall. This has a thinner muscular wall. This is a larger lumen. This is a smaller lumen. These are 2 blood vessels, large blood vessels. Remember, the mucosa is one of the places where we have those large blood vessels. And here we have a nice big vein and a nice big artery where we can actually still see some of the red blood cells inside of them. Isn't that awesome? Yep, exactly. That's what that is. That is a blood vessel, or blood vessels, plural. Here's another vein, right? Again, we can tell by the thickness of the wall. So 2 veins and an artery. That's what those are, blood vessels. And teeny bit adipose, because it's everywhere. Again, here I wanted to give you the close-up of those intestinal crypts, right? In the colon, we have these intestinal crypts that are just going to be completely lined with mucus. They tend to be very linear, aligned with that mucous membrane. It is something that is super obvious, right? And of course, they're also the crypts of Lieberkuhn, because good old Bob Lieberkuhn was the first one who described them. Good old German Bob Lieberkuhn. And so, but again, that really, really nice, obvious characteristics. Again, doesn't have the invaginations the same way. We don't have the pits and then the gland. So it is much more uniform in its structure. And so again, it's one of the ways to tell it apart from the stomach, not maybe as obvious as the muscularis. And again, obvious is what is important. All righty, questions on the large intestine? All right, from the colon, we go to something we should be familiar with, and this is the pancreas. Notice, oh cool, again, we have a nice vein and a nice artery sitting in here. Here we have a nice duct, and as we know, the pancreas is a mixed gland, so it has 2 different components to it. The majority of the pancreas are these tight, uniform, ball-shaped structures. And of course, these are what?
Acini?
Yeah, those are the pancreatic acini. Do they do the digestive function or the endocrine function?
Digestive?
Yeah, they do the digestive function. They're responsible for producing the pancreatic juice. This is where we're going to produce the bicarbonate. This is where we're going to produce the enzymes. This is where we're going to produce all those stuff. It releases it into the duct, and the duct releases it into the duodenum. Again, we're a little beyond where we were in lecture for that part, but we did talk about the pancreas and discuss it there. So that's what we have here. There's that duct that's going to lead to the main pancreatic duct and express it out. And these are the exocrine functioning acini. But we also have these islands in the stream, right? That's what they are. And again, these are the exocrine components. Right? This is where we have the alpha cells and the beta cells and the delta cells and the gamma cells that were producing our insulin and our glucagon and the other hormones that are produced from the pancreas from a exocrine standpoint. And again, they're releasing their hormones into the interstitial fluid where it gets picked up by the blood and carried away. So it is not releasing it. Oh, and conveniently enough, we have a blood vessel coming out of this pancreatic acini here. Pancreatic islet. So the acini feed into the ducts, gets expressed outside the body. The islets feed into the blood. Right? Nothing really super new here. This is just something we've seen before. What is new, and this is going to take some heavy lifting when we get to it, is the liver. I learned something new as I was exploring the liver and trying to find you some good histology. What you were looking at is the liver lobules of a pig. Because it turns out a pig's liver has extensive connective tissue between the lobules. Our liver, this connective tissue is much thinner, much more flimsy. It looks a lot more like this. Right? Notice you can barely see where some of the connective tissue is located in between of these particular structures. But what we have here is a central vein and then a lobule around it. And what we see is that really beautifully clearly here in our pig one. So I love this picture of the pig liver. Pig liver is now my new favorite thing because of these awesome connective tissue septums that separate the individual lobules. Our liver has over 1 million lobules in it. They're these kind of hexagon-shaped structures with a big, huge central vein at the center. And as we know, as we've talked about, the liver is where our magic happens, right? Where we have to process all of the blood that is coming from our digestive system, and we need to make it nutrient appropriate. Because if you've just had a meal, it is nutrient rich. We've got to make it nutrient appropriate, right? So it needs energy to be able to do that. And so it's a huge function of our liver. But part of the problem, as I just hinted at, is really it needs two things. It needs blood from the digestive system, so that it can get the rich nutrients. But as we're going to see, getting that nutrients requires energy. So the blood coming from the digestive system is rich in nutrients, but it is O2 poor. All the work the liver does requires a massive amount of oxygen. It needs oxygen. But if you remember back to the cardiovascular system, oh, and let's be specific. Hold on, let's come back up here. So blood from the digestive system is nutrient rich, nutrient poor. And what is that blood vessel that brings the blood to the liver from the digestive system again?
The hepatic portal vein.
There you go. It's the hepatic portal vein. For the liver to do its job, it needs oxygen. Luckily, there is a systemic artery that can bring that oxygen to the liver. And what was that blood vessel that brings systemic blood to the liver?
Hepatic artery.
There you go. The hepatic artery branched off of what blood vessel?
The descending aorta.
True. Okay, but so give me the pathway from the descending aorta to the liver. What's the first branch that it comes off of the descending aorta? Off of the abdominal aorta. This is a cumulative class with a cumulative final exam, right? All right. How about the celiac trunk? And that celiac trunk feeds into the common hepatic artery, which feeds into the hepatic artery. All right. We'll talk about this in class, so it's okay that you don't remember this, but we'll need to know it. So notice, we really need two blood vessels to come to our liver. We need our hepatic portal vein to come, and we need our hepatic artery to come. One's going to bring the nutrients that need to be processed, but one's going to bring the oxygen so that it can do its work. And that liver does a lot of work. If that wasn't enough, if processing all of the food and maintaining the nutrients and doing all those other many things it does to help to maintain the blood isn't enough, your liver also, on top of everything else, as a hobby on the side, produces bile. And that bile is used, as we talked about, to emulsify fat. And so it needs to not only get blood from two different sources, but it needs to be able to release that bile either into your digestive system or send it to the gallbladder to be stored. So we also need a bile ducts, some bile ducts as well. So if you notice, there are really three different vessels that need to be associated with this liver for it to do its job. And conveniently enough, these three things are known as the hepatic triad or the hepatic portal triad. We don't necessarily see them in this illustration, although we do see a little bit of it. I will show you where they are, and they are going to be located. So if you think of this hexagon shape, here at the corners of our hexagon, so kind of as if you notice at the divisions of the connective tissue septums, in these regions is where you're going to see that hepatic portal triad. All right. Now I'm going to cheat. Let me erase these since I did them, but I'm going to see if I can leave the words there on the side because I don't remember what the next picture looks like, but it might be useful. Here is that central vein. Here are the capillaries, the sinusoid capillaries, where these things are coming through. And here we can see some of the structures that are going to be located here. Notice here we see part of the hepatic duct and a blood vessel and stuff like that. But here I've got an even better view. This is what I wanted to show you. This is the last important thing. I know we're a little over time, but that's okay. I figured this was important. This is the last thing we needed to look at, but it's the trickiest part of the liver. So it's the part that I wanted to make sure that we were able to do, because you are responsible for identifying these things histologically. The hepatic portal triad. 3 things. Now, I'll make it easy for you by circling the 3 things first. This is thing 1. This is thing 2. And this is thing 3. So let's number them. 1, 2, 3. 3 things. Which of our 3 things here is the hepatic portal vein?
Number 2,
There you go. How do you know?
because it has a thinner wall.
And a huge lumen. Thinner wall and a huger lumen. Typically, the hepatic portal vein is going to be the largest of the 3 structures. Which one is going to be the hepatic artery or a branch? And again, remember, this is not the only hepatic portal vein. This is a branch. So when we're talking about these are all branches of, right? Because there are millions of lobules, so there are millions of branches of. But you're right, this is the branch of the hepatic portal vein. Which one of these is a branch of the hepatic artery?
Number 1.
Number 1, there you go. It's got blood in it. That's always a dead giveaway that it has red blood cells. But notice it has a thicker muscular wall. Notice also, again, remember, blood vessels are lined by simple squamous epithelial tissue. Often the nuclei of that simple squamous epithelial tissue are so large that they stick out beyond it. And so you can kind of see the linear organization of those simple columnar epithelial tissue. Whereas bile ducts are typically made up of cuboidal or columnar cells. And lo and behold, what do these look like over here?
Simple cuboidal.
Simple cuboidal cells. So notice here, object 3 is a branch of the bile duct. Right? Notice we have another duct right here. We can see those cuboidal cells up in a circle. So we also see a nice one there as well. All right, so we are going to talk, this is actually where we're going to pick up in class on Monday. We will be talking about the liver and the pancreas. Pancreas we have some familiarity with, but this I think is the biggest challenge, being able to histologically distinguish these 3 things. But if you really focus on those key components, as you mentioned, the veins always have a thinner wall and a large lumen. Here we can see the nice pink of the muscularis of our artery, so it's going to be thicker. And here is our bile duct. The hepatocytes are basically all the rest of these nuclei we see. So the rest of the majority, and in fact, I think we'll see this. So any questions on this? I want to go back to the previous slide. So before I change this, any questions on these 3 things? All right, let's go ahead and clear this then and go back to the previous slide, and we'll get a better sense of answering your question. Notice, and again, here we can see a duct. Notice it's cuboidal in its shape, so that's why we're able to see that there, and some blood vessels here. Notice there's that big central artery. All of these cells, they're kind of lined up as rows, and I'll kind of emphasize this. This is kind of an oversimplification, but we have these. I can't rotate these at all. These are horrible. I'll just do it as lines. Let's undo that, and we'll use my highlighter, and we'll make it thick. So what we have are rows of basically these hepatocytes that radiate out from the central vein, or really radiate into the central vein. So we have these lines of hepatocytes. These are the cells of the liver that is doing their job, and these spaces that we see in between, let's change the color to emphasize this, these spaces we see in between, these are the sinusoid capillaries. Remember, we talked about how the liver is one of those places where we need to be able to move big things in and out of our blood supply. And so in these spaces in between, we have these nice big sinusoid cavities, capillaries that are going to allow that. Your book has some great illustrations that do a really nice job of looking this. So we'll start with the illustrations and then maybe come back to the histology to see that it makes sense. But so yeah, the hepatocytes, they're what's responsible. So that way, basically the two bloods mix together. The oxygen-rich blood from the hepatic artery, the oxygen-poor but nutrient-rich blood from the hepatic portal vein feed into these sinusoid capillaries. And basically what happens is the hepatocyte uses the oxygen to process the nutrients. And so when the blood reaches the central vein, now it is nutrient appropriate. Of course, it's now oxygen poor, which is why it gets fed out into the hepatic vein, which goes into the inferior vena cava and goes into the right atrium of the heart. And there you go. That is your histology list. Any questions on any of that? Stunned silence. Excellent. Just what I love from a class. All righty. Well, we went a little bit over, but like I said, the advantage of this was we got a chance to go through everything. Hopefully this helps. Again, use this to help you to be successful on the exam. As you can see, there is a wide diversity of histology on this exam, but it is diverse. Most things don't look like other things, so it should hopefully be pretty easy to distinguish them and tell them apart. I would say, well, obviously I haven't done it online before, but in the classroom when we do this, we sometimes do it station-based. We sometimes do it as a PowerPoint presentation, but I would say easily a third of this exam could be histology. So make sure you're familiar with this. And again, to help benefit you on that, I am encouraging you to do the lab work, to do the histology, and I will give you extra credit for doing that. All right. Any other questions? Is there a picture of the lobule of the pancreas? No. I think the picture of the pancreas I have is too high of a magnification. But what I will tell you is if you look at a lower magnification view, well, I'll cheat. If you look at a lower magnification view of the pancreas, what you'll see is like the liver, there are going to be connective tissue septums that separate individual lobules. That is something that the liver and the pancreas have in common. This particular view doesn't show any of the connective tissue, but if you look at this from a lower magnification, or even if we were in a different view, what you might see is you might see this big, huge swath of connective tissue that went through the center of this, and that would be that connective tissue septum that would separate the individual lobules of the pancreas. The lobules of the liver have that kind of distinct hepatic shape to them, hexagon shape to them, whereas there is no distinct shape to the pancreatic lobules. They'll just be divided by connective tissue. All right, good question. Any others? Give you guys one last chance. All right, then I'm going to go ahead and stop the recording. Then I'll get this I'll get this posted onto YouTube today. For those of you that miss it and are listening to this again, because we didn't get a chance to talk about the assignment, I'm not going to hold you responsible for it on Monday. But that does mean that it's due before the exam. And remember, the exam starts at 7 o'clock on Wednesday. So make sure you get it done before then. All right. Good luck. If you have any more questions, please feel free to reach out. Otherwise, you guys have a great weekend. Study hard. Remember, this is your study weekend for the exam. Take advantage of that. Start looking ahead at the things we're going to talk about. Remember, we're behind on lecture. So we got to cram on Monday to get that in. So don't let Monday be the first day you look at this stuff. All right. Be safe. Take care. Have a good weekend.